In this talk, I will discuss how the connectivity structure of biological neural networks shapes their dynamics. In particular, I'll try to explore which are the critical features of the connectivity of neurons that shapes their dynamics. This work is done with Simacho Mengiste, Sebastian Spreitzer, Shriyan Fawaz. Part of the motivation for this talk comes from this commentary that appeared in Cell last year. Um, the commentary said that we need to chart out the connectome of a mouse in order to find out the structures of long-term memory, to better describe the neuropathology of brain disorders, and very curiously to design non-biological thinking systems, because they think that knowing the connectome of a mouse can give us blueprints for cognitive computing systems, data efficient learning, and energy efficient computing. And together, this is what perhaps constitutes the mind of a mouse. So what is actually mind? I'm very much inspired by Warren McCulloch's article, Why the Mind is in the Head, when I think of the mind. To me now, mind seems like what we could call as ideas and purposes. These are the things an agent needs to survive in an environment, to interact with the environment. And that requires finding invariances and regularities. And to do that, this agent needs certain algorithms. These algorithms, to me, seem like a better definition of what we may call as mind. Then there is the brain. Brain is the material stuff. It's made up of all these chemical, biological stuff. And it has its physiology, its development, and the fact that so many neurons are interacting with each other at different time and spatial scales. The brain has its own dynamics. Now, obviously, we know this, that the brain hosts the mind. That means that somehow the brain activity dynamics holds clue for the algorithms it is running. And it makes sense that if we knew something more about the brain, perhaps we can unravel the algorithms the brain is using, which of course are very energy efficient and work for very sparse data as well. So in this talk, I'm going to explore a little bit about the structure of the brain, particularly the connectivity structure of the brain, staying pretty much in the middle layer of the description of the brain, if you wish. The plan is that I'm going to review the known data about the connectivity of neurons at different spatial scales. Then we will delve into briefly non-spatial models of the brain connectivity, and finally go into spatial networks and briefly touch upon heterogeneity of neurons as well. So when we look at a gross scale, um, there are certain brain regions where you can easily identify, there are anatomical landmarks to identify subregions that are connected to other subregions. In hippocampus, basal ganglia, and amygdala, we can clearly identify different subregions. And this kind of gives an impression that these structures are organized as network of networks. For instance, in the hippocampus, we have CA3, which is a recurrent network, but CA3 itself is a part of a more elaborate macroscopic network, if you wish, where it receives input from dentate gyrus and sends its output to CA2 and CA1. Same thing can be said about basal ganglia, amygdala. For the neocortex, it's not as easy, but still it's possible to segregate different parts of the neocortex based on their functional specialization, as was done for vision, for instance. Nowadays, we have better technology. We don't have to rely on these specific anatomical landmarks or functional specialization. At the Allen Brain Institute, this is quite remarkable that they are at different spatial scales able to characterize connectivity of different parts of the brain with each other. Here, this is an example where they injected tracers in different parts of the brain and estimated how the tracer goes from one area to another. There's no reason we need to remember this connectivity. All I'm using it to highlight the fact that it's not random, it's highly structured. There are connections which are within one hemisphere, but there are connections that cross over. But even within one hemisphere, there's a large degree of structure that exists. In this paper by Harris, they argued that there seemed to be some sort of hierarchical arrangement that may be a bit difficult to defend, but still there is seemingly a flow of activity even at the structural level, it seems possible to infer that it goes from sensory to higher cognitive areas. There's also a temptation to think about that whether this particular connectivity has special properties. For instance, is it scale-free, small world, and so on. And we'll think about it in the next few slides, whether it means anything or not. So that's the situation at the mesoscale. At mesoscale in the neocortex, we can also see another kind of organization, and that is that neurons seem to be organized as layers. In mammals, there are 6 layers. In birds, they are not there. So at some point, it was believed that this may be something very crucial for higher cognitive function. But since birds also have cognitive abilities, so perhaps there's no such special. Maybe the layers are not as special as we might have imagined. Within a layer, if we look at short distances, it turns out that average connectivity of neurons depends on layers where they are. And the connection probability decreases as a function of distance. In this case, for instance, if a neuron is sitting in layer 2, 3, its connectivity will decrease as a function of distance, both for excitation and inhibition. Seems like in XY plane, at least, they're pretty much balanced with some mismatches. In the z-axis, we can see more differences, but these differences partly are due to the different density of neurons in the layers. In this case, this is a summary data where the neurons were not separated into different classes. If we do separate neurons in classes, then Tolia's lab published this beautiful data where they showed that different types of cells connect to each other with different probabilities. And if you look at just this connectivity matrix, it looks pretty non-random. But they argued that if we correct this matrix for the size of the neurons, then the connectivity is pretty much distance dependent and random. So we need to take into account the specific shapes of the neurons. There's another structure at this scale that, first of all, just a quick reminder that neurons seem to be, at least in certain mammals and primates particularly, neurons are organized in early visual system in these pinwheel patterns where there are these domains where neurons seem to have a preference for certain kind of orientation. And it turns out that if you look at the connectivity of neurons within an orientation domain, they not only make local connections, but they also make faraway connections. These are called patchy connections. And these patchy connections are made preferentially with other neurons that have similar stimulus specificity. Here we can see the nature of patchiness much more clearly. The tracer was injected at the center, and you can see how all these neurons make patches far away. So this is another feature of the connectivity in the brain, that there are long-range patchy connections, and there's a structure to that. If we zoom in further, then today we have technology that we can record many cells simultaneously, and we can look into what happens to the responses in different neurons as one of the neuron is activated. In this case, they could make 8 patches simultaneously. People can do also 12 patches, very tedious, time-consuming work. What this has revealed is that if we take the Erdos-Renyi type random network as a benchmark, then cortical networks are highly non-random. And this could be demonstrated by counting the probability of finding such motifs, let's say of size 3, where you ask a question, how likely is it to find a set of 3 neurons which are bidirectionally coupled to each other, or in this form, or so on. At 3 neuron level, we can find 16 different motifs, and it turns out that these higher order motifs are much more likely in the cortical connectivity than in a Erdos-Renyi type network. And these days, it's possible to do it in other areas as well, where you find that motifs of, let's say, 6, 7, 8 cells, they are much more prevalent in the brain than in Erdos-Renyi type networks. And now this has been recently shown also for the C3 as well, where these convergent divergent motifs are much more common than expected from Erdos-Renyi type networks. So there is some structure there as well. In the neocortex, there's another aspect which relates to this idea of motive. Idea of motive has been very old for neocortex. In fact, since long it has been known that the neocortex could be thought of as a collection of many mini columns. This idea was initially proposed by Mountcastle, who saw that vertically, if you look at neurons, tends to have similar stimulus preference. And then he thought that this could be considered as if it were a column. And then many such columns are put together to construct the neocortex. In the neocortex, now we know much more detail about the connectivity. In particular, they have identified 4 different types of interneurons, and their connectivity scheme differs whether the neurons are sitting in the superficial layers or in the deep layer. But together, this is highly non-random, and there are some interesting connections which give counterintuitive dynamical properties. This particular structure of how the activity goes from, let's say, sensory input layer, layer 4 to layer 2, 3, and back to 5, how this could be a canonical microcircuit to mix forward input with backward input, meaning to do some sort of Bayesian inference where your input interacts with your top-down expectations. So very quickly now, that means that brain is definitely not a random network, at least not in the sense of an Adosh-Renyi type network. And there are several non-random features that exist, and they exist without any learning or any training. And this to me tells that there are some generative rules that the brain uses to wire itself up. And one could argue now that if we think that the secret of the cognitive abilities of the brain is in the connectivity of the brain, then most likely these non-random features are actually important key to find out the function, or let's say mind. So essentially, in this commentary, the claim is that if we know the structure of the brain, we will know the mind, or function at least. Now, this issue of function is very tricky. Now, what is the function? Function really depends on many different things, not just the object that you have in your hands. Taking a trivial example, for instance, what's the function of a hand? Well, the function of the hand depends on the person whom it belongs to. Similarly, function of a network depends on where it is embedded in. It's not a unique property that exists without the embodiment of this network in the environment. And this idea was very nicely demonstrated by Hopfield and Tank in their seminal paper in 1986, where they introduced the Hopfield network. And I feel that everybody remembers Hopfield networks, but they conveniently forget the first part of the paper. Very first part of the paper has this figure where Hopfield and Tank pose a challenge to the reader that here is a network of 7 neurons, 4 excitatory and 3 inhibitory. They give us the connectivity of this network, and the task is to find out what is this network actually doing. And it turns out that it's highly non-trivial to come up with a function for this network. At least for such small networks, it may be possible that after looking at this drawing for a long time, you may come up with the notion that maybe it is doing analog to digital conversion from analog signal to a 4-bit digital output. But that you can perhaps know only because you know that A to D conversion is meaningful. If you didn't have any background in digital electronics, it wouldn't even make sense to think about this. So the claim that was made is that it's not really possible to talk about function of a network by just focusing on the activity or connectivity of this network. We don't really have any principled approach that we can scale the notion that if we can derive the function for a small network, that we can also do it for a larger network. There's no such approach. Only thing we have is perhaps the framework suggested by David Marr, the 3 levels of problem, algorithm, and implementation. But really, we have almost nothing to make this approach that if I know the connectome, I can talk about the function. Hopfield mentioned this problem once again because perhaps he realized that nobody got the message. In 2000, end, he published this paper in PNAS, and there was a follow-up. In the first paper, they posed again a similar challenge, that what's the network, and in the next paper, they revealed the network. So again, from 86 to 2000, there was no real progress on this question. We revisited this question and made a challenge where there are networks, you can interact with them and try to find out the function of those. And more recently, Conrad Coding's group, they really went into this and said that can we really use our approaches to even understand a microprocessor? So it seems that it's a very much wishful thinking at this moment to say that if I know the connectome and I use our contemporary neuroscientific approaches, that we can talk about the function. And this has been articulated very, very nicely by Alex Gomez-Marin in this recent commentary that was published in Neuro. So then what is it good for that we know the connectome? And clearly we know that connectivity is important for the brain function. And as Hochfeld said, and has been repeated many times since, that we can't really talk about function just knowing about the connectivity. So what is connectivity good for? In their paper itself in 86, they argued that there is something else we can do. We can define the dynamics of this network. And in this case, they were focusing on the energy states and looking into the minimum energy conditions. And that, based on that, they argued, well, we could use such a network for optimization problem. So we have some principled approach where we can relate the connectivity to the dynamics. That's definitely possible. And that's what I would like to explore in this talk, what kind of dynamics and computations might exist in complex network structures. And other way around, that how can we relate the structure to the activity dynamics? If we have this understanding, then at least we can guide our search for connectomes and not measure everything, but be very specific in what we want to extract. So there are two reasons I want to say why dynamics is important. One is that dynamics is, in some sense, it gives us some very basic objects that can be used for computing. For instance, networks, they have self-sustained activity, and that self-sustained activity which persists after the stimulus is gone could be used for instance, working memory or movement hold circuits, etc. Similarly, dynamics gives the network's ability to amplify, attenuate, or filter signals. We can generate spatial temporal sequences and create contextual switching. And at least in the Hab's vision, those contextual switching of sequences could already form the basis of information processing. But even if we say that, well, this is perhaps not the way how the brain works, then the claim or the position I would like to take is that if dynamics is not the computation, at least it is the substrate of computation. Then computation must exist in a certain dynamical environment, and almost always any computation should not destabilize the network or create bifurcations. So in some sense, dynamics gives the preconditions to process information and puts boundary on what can be computed and what cannot be computed. So with this, I would like to explore the dynamics, how structure relates to dynamics. And when we think of the structure, very obvious choices come to mind, obviously inspired from the known structure of the brain. Feed forward. Well, brain is not really a feed forward network, but still, since there are some hierarchies, we can treat it as if it's a feed forward network over short time scales. But really, it's a recurrent network. So we start simple with Addo-Shreni type recurrent networks, then locally connected recurrent networks, and finally also with patches. When we are thinking of network dynamics, we also need to consider what the network is composed of, whether it has only inhibitory neurons, for instance, in the striatum or in some parts of amygdala, or is it like the neocortex, where there are both E and I type of neurons and they are mutually coupled. And in between, we have the CA1 type of networks where they're both E and I type of neurons, but E and E neurons don't really make connections. These kind of networks have been studied a lot in last 2 decades or so. in various forms. So this talk is not really a review of this, but definitely my ideas have been inspired by all these investigations. So let's start with non-spatial networks and also define what we mean by dynamics here. So to define the dynamics, we can focus on individual neuron properties as a part of the network and the population properties. Individual neuron properties can be very nicely captured by their firing rate. That would be number of dots in each row. So whenever a neuron spikes, there's a dot. This is a raster display. A neuron can spike in a regular or irregular fashion, like a clock or a Poisson neuron. In the form of population, the population can oscillate and or synchronize. So this way, with these 4 parameters, we can capture both individual and population properties. So I'm going to restrict my discussion largely to these. For a random Edo-Shreni type of network with E and I neurons, we know now for quite some time that the balance of excitation and inhibition is very crucial. And by changing the ratio of excitation to inhibition, we can tune the network to operate in the so-called asynchronous irregular state, synchronous irregular state, or synchronous regular state. And clearly these transitions between the states are not discrete, but there is a continuum, and that continuum is shaped by the balance of excitation and inhibition. On x-axis, the ratio of inhibition to excitation, on y-axis, the external excitation. So there are regions where there is AI activity, and there are regions where there's more or less pathological synchronous regular state, and in between you have interesting SI states where neurons tend to synchronize but still spike in an irregular manner. So for this, a lot of analytical simulation work indicates that the key is this balance, the factor G here. Now, what happens if we go away from Edo-Shreni networks and look into other topologies, which are very popular, for instance, small world, where neurons make shortcuts such that the average path length is very small, and still there are clusters, or scale-free, where there's a wide distribution of degree of neurons. So what we did is that we took these different topologies, we set up a simulation, and we adjusted parameters in such a way that in our baseline state, all of them are in more or less similar dynamical state. Now, this is not difficult. You have to fiddle with the parameters a little bit, but mostly for these 3 topologies, it's possible to tune them in the similar activity state. So first question we wanted to ask is that, well, does this topology matter? And if you look at this picture here, you would say that, well, it doesn't really matter. However, a counterclaim would be that here we have tuned everything so that they look the same. So what happens? How do we ask this question that does the topology matter? So we came up with this idea that let's perturb the topology. If the network is sensitive to perturbations in topology, then we will be able to find out if it matters or not. So for perturbing, what we thought is that, well, we can do 2 things together. We can introduce a perturbation and also ask a question about neurodegeneration or synapse pruning in the brain. So we start with a network which is 10% connected and progressively remove synapses. And to remove synapses, we use 5 different approaches. In one case, we remove synapses randomly. In another case, based on the degree of the neuron. And a very complicated strategy is that where we look into the matching set, and depending on that, we remove synapses. And what you see here is that for an Edo-Shreni network, it doesn't really matter the way you remove synapses. Firing rate starts at low, and as you prune synapses, firing rate increases. Now, this is not really a surprise because we have tuned this model to operate in the so-called inhibition-dominated regime. And as we remove synapses, inhibition is released and firing rates, they start to grow. But the surprise is that the same trend is observed in also small world and scale-free networks. Qualitatively, there's hardly any difference in these. So at least at the level of firing rates and synapse pruning, we know that topology doesn't matter. And we confirmed it also for neuron degeneration, not just synapses, and pretty much the same pattern is observed. There are 2 pruning strategies which are a little bit more robust in scale-free models, where the increase in firing rate is not as pronounced, but the trend is still the same. We check this also for spike time irregularity and synchrony in the network. And it's almost indistinguishable how different types of networks behave for different pruning strategies. There are pretty much only two exceptions that we notice. One is in this synchrony in scale-free models where we are removing neurons, and the exceptions appear when we are removing neurons based on their out degree or in degree. And obviously, this is a very special perturbation because we don't really destroy the network in this case, so they can increase synchronization given that inhibition has been released from the network. So to us, it was a big surprise that even though we start with different topologies, we prune them either by synapse removal or neuron removal and different strategies, yet in all possible cases, we get basically the same qualitative behavior. And this makes us think that maybe topology is not really an important property of these networks. But then what really matters? So what Seema did then is that he did a regression between different structural measures, effective synaptic weight, which is essentially EI balance, its variance, shared connectivity, number of connections, the distribution width of the connections, spectral radius, EI balance, the ratio of excitation to inhibition, number of neurons, and connection density. And in the dynamics, he was again looking into the 5 properties that we are interested in related to firing rates, regularities, and synchrony. And what he finds is, very curiously, that the best description of the network dynamics can be obtained just by looking into the effective synaptic weight, which is really the sum of total excitatory and inhibitory neurons, inputs. And this you can appreciate much better here, where he plotted ESW against the CV or the population synchronization and firing rate. It's a beautiful correlation that we can appreciate here. So ESW is actually a very interesting property because this you can measure simply by patching a neuron. So it makes the task very easy. But the question is that what happened to the topology? So it turned out that topology is really a very fragile property as we start to prune the network either by synapses or not. Only in these red regions, you find that the topology is preserved. As soon as we start to disturb these networks, they essentially lose their topology. And then it's not really a surprise that topology doesn't turn out to be an important parameter for the dynamics. So something that we thought is very robust, or something that makes robust the networks by itself is not robust, meaning scale-free, small world topologies, they are extremely robust, meaning the network retains its structure. However, the topology changes. And this is important for the brain because the brain is constantly undergoing developmental changes, learning related changes. So topology most likely cannot be maintained because after all, there are only few algorithms that we know how we can wire a network as small world or scale free. So any learning related change, developmental changes, etc., will perturb the topology massively. So in some sense, it's maybe a good news that topology is not relevant property of the network. So this is true now for non-spatial networks. And obviously here we have a very restricted definition of effective synaptic strength, which turns out to be the best descriptor of the dynamics. We have ignored the timescales and maybe in some future work that we can explore. But changing gears to now spatial networks, because after all, the connectivity in the brain is spatially constrained. We have very useful tools to study activity dynamics in spatial networks, and they are they're called neural field equations, which were pretty much inspired from the work of Alan Turing, where he talked about how different chemicals interact to form patterns. So here in the brain, we have 2 types of interactions, excitatory and inhibitory, and if they have space constants, depending on how far they connect to, that pretty much creates the same situation as Turing was exploring. And this is something we can write down as a neural field equation where we can describe the activity of a neuron at some location X as a function of time. And the key variable here, of course, is the connectivity kernel, which tells us that how this neuron wires or in what way the connectivity depends on the distance between neurons. Now, We can solve these PDEs in restricted conditions. For instance, we can talk about what will happen to the stationary state if we perturb it locally. And this simple perturbation experiment gives a very nice insight, because it turns out it's easier to solve these equations in Fourier domain. And in Fourier domain, to know whether this network is going to be stable or unstable to the perturbation, it really depends on the eigenvalues, and those eigenvalues depend on the Fourier transform of the connectivity kernel. So the summary statement of this analysis is that we need to know the Fourier transform of the connectivity kernel. And if we are interested in spatial patterns in the network, then the connectivity kernel should be non-monotonic. This is something we can appreciate here, where we have plotted this equation, solutions of this equation for different parameters. And if we have a monotonically decaying connectivity, for instance, a Gaussian kernel, in that case, the lambda values, they monotonically decay. And this indicates that in the network, there's no real spatial structure. So this pretty much tells you the situation about spatial oscillations in the network. And if we change the connectivity from Gaussian to, let's say, non-monotonic, for instance, a Mexican hat shape, which easily arises as we sum up excitation and inhibition together, then it turns out that there's a possibility to form a spatial periodic pattern. And the periodicity, of course, will depend on the properties of the connectivity kernel. So this whole the analysis gives us a heuristic that whenever the connectivity kernel is non-monotonic, we should expect spatial patterns. And if it is not, then we should expect homogeneous activity. And this we really confirmed in an inhibitory network. This is a question we can only ask in an inhibitory network, because if we had both E and I, then the effective connectivity could become non-monotonic. So in an inhibitory network, if we have Gaussian connectivity profile, it just decays as a function of the distance, then no matter what you try, if you drive the network with homogeneous input, it will never form any structures. By contrast, if the connectivity is like this donut shape, where we have now a non-monotonicity, and we model this as a gamma function, therefore, high input, the network settles into these localized bumps of activity. And you can better appreciate it in these spatial average plots, where we can vary now the properties of the donut by changing the parameters of the gamma function, shape and scale. You can get different kind of patterns. But essentially what is happening is that as you change the scale, you're just changing the frequency of these patterns, nothing really much changing. So the heuristic is correct that non-monotonic connectivity is important and crucial for spatial patterns. There's a lot of other work in this, how we can adjust the connectivity kernel to find out many different spatial temporal patterns, traveling waves, standing waves, multiple bumps, and so on. So there it also becomes important what are the delays in the connections. One thing that was not clear much is the role of the patches, whether the patches are relevant or not. And now, since we have this heuristic, it suggests that if we had patches are going to create another non-monotonicity, and that should create some qualitative change in the network. But obviously, just looking at this figure, we can also say that it really matters how big is this non-monotonicity and where is it located. If, for instance, the patch is very close, this may not really create much of non-monotonic structure, and therefore it may not have an effect. So we had some heuristic that depending on the location of the patch, we can increase the number of bumps, and bump should become unstable, and it should create traveling waves. And this we checked analytically first placing the bump at different locations and look at the eigenvalue spectrum. And you can really see that as you move closer and closer to the main lobe of the bump, the frequency of spatial pattern decreases, meaning that we should get different number of bumps depending on where the patch is placed. And this we can appreciate here, that if you're closer, for instance, to the bump, then the frequency of patterns is much higher, which we can see in this plot here, that when the patch is closer, then we follow this blue line here, and which has a peak at 9 or so. So essentially, we are going to increase number of bumps depending on the size of the network. On the other hand, if the patch is far away, we don't really change number of bumps, but they move relatively fast. This is something you can appreciate better in these movies, which Fawaz made. He places all the neurons at the center, and as they become active, they jump out. So you can see the three lobe appearing when the patch is slightly far away from the main lobe, as opposed to when the patch is sitting closer. Now we predicted that there should be more bumps, but they should move slowly, and that's what exactly comes out here, that there are pretty much 5 or 7 lobes we can see, and they move very slowly. So right, the patches are important in the sense that they introduce a non-monotonicity and they tend to create these traveling waves and multiple bump solutions. Now, I'm not claiming that this is the model of traveling waves in the brain, but we do see traveling waves in the brain, and possibly these patches may contribute to that. Now, what about heterogeneities? So far, what we have seen is either non-spatial networks or spatial networks, but both of them were homogeneous. Connectivity can be highly heterogeneous in the brain. So what is the role of the inhomogeneity, and when do those inhomogeneities matter? To ask this question, what we did is that we had a spatially connected network where we changed the direction preference of this neuron so that they tend to connect in a given direction. And only a small fraction of neural projections go in that direction. Otherwise, it's pretty much isotropic. So we faced a problem here that how do you distribute this preferred angle for each neuron? In one limiting case, it could be that all the neurons are projecting in the same direction. In other case, they could be all random. And in between, one can imagine that neighboring neurons tend to project in similar direction. And the amount of change that we bring in the connectivity by shifting or by creating this preference is not so much. It's only changing by 1 or 2% at best. So what comes out when we have this kind of connectivity? I think it's better to appreciate it, or we can better appreciate it in movies. So when we have no preference, then we get this multiple bump solution. They are stationary. And as we introduce some randomness in the preferred connections, nothing really changes in the network. However, if we now make all the neurons connect in a given direction, so that everybody is correlated in their preferred direction, you get these nice traveling waves. And an interesting scenario appears where neurons have neighboring neurons have similar preferred direction, and far away doesn't matter. There you get these multiple sequences that travel in different directions in different parts of the network. And this kind of behavior is not really, so what Sebastian did was that he only looked into the E2E connectivity, and that was made anisotropic and spatially correlated. We can do the same thing for E2E connections or I2E connections. In each case, you will see these sequences moving. So in this case, Shreya changed the connectivity asymmetry between E and I connections. So there also you have this nice flow of sequences. Similarly, when you do it in an II case, that also works out. So it doesn't really matter in which of these 4 connections you introduce the correlated asymmetry, we always end up with spatiotemporal sequences as long as there is some correlation in their preferred connectivity. And this kind of gives us an indication that maybe this is the possible neural substrate or connectivity substrate which underlies emergence of sequential activity in neural networks, often experimentally seen and displayed pretty much like in these pictures here. Now, this correlation, the spatial correlation, that's important. In one case, it gives us traveling waves. In other extreme, when everybody has a different preferred direction, it gives us standing bumps. So there is just a continuum here. If you have more local correlation in the preferred connectivity of neurons, you get bigger sequences, fewer sequences. So it's kind of just a continuum. But what is the key is that there should be some spatial correlation in the degree of asymmetry of connectivity. This kind of connectivity, we have wired it, and it's not really possible to measure it right now, whether neighboring neurons have such preferred or similar dendritic axonal arbors. But there are other ways how you can get something similar in a more functional sense as well. So recently it has become possible to measure the spatial and temporal profile of neuromodulators, in this case dopamine, using optical sensors. And it turns out that both the receptor density as well as the release pattern of neuromodulators uh, uh, has a spatiotemporal structure. It's not homogeneous, it has patchiness. And in fact, even the release pattern of the neuromodulators seem to follow some kind of wave-like structure, which changes its direction and orientation depending on the task. So neuromodulators are constantly being released in different behavioral conditions as animals are getting rewarded or not. That means these modulatory substances have a chance to shape the structure, and the shaping of the structure will depend on the release pattern of the modulator itself. So here's a schematic where you start with a completely homogeneous network, and then comes this cloud of dopamine because of whatever task condition was there. Neurons that end up, or synapses that end up in that region, they will get strengthened. So essentially, this network will be restructured according to the gradient, spatial and temporal gradient of neuromodulators. And thereby you can create some sort of sequential flow. And this flow will of course not be permanent, because this will change depending on the new spatiotemporal pattern of the dopamine. So this kind of gives us a very nice, powerful control over functional circuits in the brain, using neuromodulators as kind of gearboxes of the system. So this also goes back to this whole idea that connectome is important. Of course it is important, but such neuromodulatory substances, they have this ability to restructure the whole connectivity and drive it into a different functional regime, something we perhaps will not be able to deduce from connectome alone. So the idea then is that in spatial networks, what matters is the shape of the connectivity. Anything that introduces uh, uh, asymmetries or anything that introduces non-monotonicity in the connectivity kernel is crucial. And those non-monotonicity could arise because of patchiness, but also dynamically controlled through neuromodulators. So just to then summarize, what we have seen here is so far that for as far as the dynamics of biological neural networks is concerned, there are certain features that are relevant. For instance, the non-monotonicity of connectivity kernel, whether it is generated by hardwiring the neurons or dynamically controlled by the neuromodulators. What doesn't matter is the topology. And why should topology matter? If we really summarize this, the key here is that whatever breaks the symmetry in the network, and symmetry now of EI balance, that parameter becomes crucial. In these random networks with scale-free small world topology, they are all homogeneous networks. So even if we are changing things, we are changing them homogeneously, and therefore we are not really introducing any asymmetry or any symmetry breaking in the EI balance, as opposed to other changes like this non-monotonic kernel shape, or the way neuromodulators can dynamically change the effective connectivity. They introduce spatial asymmetries. They disturb the EI landscape in the network, and therefore they are important. Now, obviously, one question emerges that what about motifs now? Whether motifs are relevant or not? Well, they are relevant only again in this context that how many motifs are present in the network. Um, this motif, we know that if we create a structure based on the cortical column motif, it has very interesting properties. But note that we are studying the whole, the networks which have nothing but this motif repeating everywhere. And in that case, it has very interesting nonlinear properties, hysteresis, and so on. But the question is that in a big network, how many such motifs should be there to generate some meaningful difference? And that, of course, needs to be characterized. My guess is that the way we have a continuum in the spatial correlation of anisotropies, I think there should be some similar continuum as well. But in any case, the point is that there is a lot of theoretical work that has been done about relating the structure of networks to the activity dynamics. And that reveals some properties that are important. For instance, the features of connectivity which have the tendency to disturb the EI balance landscape. Those are the ones that we should be measuring, and not every random detail that might exist in the biology of the brain. Yeah, thank you so much.